Hello, guys. So for this one, we're going to do a summary for protein synthesis. Remember, protein synthesis is where you make protein. Synthesis means to make protein, obviously. It is the structure used for structures in the body. So it is one of the macromolecules used for structure. So there's two processes in protein synthesis. The first one is transcription, where the DNA is unzipped using helicase, which is an enzyme that cuts the molecule in half. And then a copy of the DNA is created using the base sequences to turn it into mRNA so that the DNA can leave the nucleus. Once the mRNA leaves the nucleus, it is called translation from that point onward. And translation is the process of taking that mRNA and turning it into amino acids. So what's going to happen is that mRNA is going to be read by a ribosome, and then tRNA is going to bring whatever that ribosome tells it to bring, and it brings it amino acids to add to a chain. So the ribosome will take that amino acid, add it to the chain. That tRNA will then leave, and a new one will come, and this will continue until a new polypeptide chain is created. As soon as it is done being made, that polypeptide chain, it'll break off and it'll twist into whatever shape the DNA was going to instruct it to be. And that's how you make that protein. Remember, proteins have a very specific shape that they need to be in order to have their function. So, genes. Genes are portions of DNA. Each gene is going to code for a protein. And so, let's say this whole strand is DNA. This portion of the DNA will be a gene. And what it does is it codes for one protein. And that is what the RNA is going to be. Okay, so RNA, the mRNA, when it's made, is going to be one gene. It's not going to do multiple genes at a time. It's going to make a copy of one gene, leave the nucleus, and go translate that gene into a protein. Okay, so one gene is going to be done at a time by the mRNA. Now, when cells read these genes, not all of them are used. So notice we just used this section. We didn't use this section. We didn't use this section because this may not apply to our cell. And so genes are turned on and off, done by like hormones will tell the cell which genes it actually needs. And this is called gene expression. So when let's say this is a blood cell and there's genes for hair color and eye color here. So this is, let's say, eye color. That blood cell doesn't need this gene, and so it'll be turned off. It won't be expressed. But this is going to be for the shape of the cell and building its cell membrane, and so it does need this gene, and so I'll use that one. So cells specialize or differentiate based off of which genes are turned on or off, or which genes are expressed, and so this portion is very important in determining that process. Finally, you have mutations, which is going to be errors in the gene sequence. Remember that you can have base pairs lined up in a certain order. Let's say like below. A mutation is an error in this sequence. You can have a point mutation in which of this group, only one base pair is removed, or you can add a base pair to the end, which is called insertion. So you can have insertion, you can have deletion, which is deleting something, or you can take a base pair, say this G, and change it into a C, and that is called substitution. So those are all point mutations. Deleting it, substituting it, or adding on insertion. Insertion. This is substitution. And then this is delete. And those are mutations. So that's what happens in a genetic code that can cause these errors. So what a mutation is will be on the test. What you also need to know is that mutations will not be passed on unless they occur in sex cells. So if this mutation occurs in a sex cell, it will be passed on to the offspring. Otherwise, it will not. And this is the only way that a mutation will be passed on. If it occurs in sex cells. If it happens in your body cells, your somatic cells, it will not be passed on. Only in the sex cells. That will be on the test. Okay, you need to know that information. It's very important to know. Mutations only pass on if they're from the sex cells. These are called mutations. So you can also have frame shift mutations or chromosomal mutations where you can have an entire section of a chromosome that is going to be incorrect. So instead of a gene, this entire portion can be incorrect, which is multiple genes, and that's called a chromosomal or frame shift mutation. Okay? And those are big. Those can cause major issues, cause things like cancer. And these mutations can be from either chemicals or radiation or just, random. But what the good thing is it does cause genetic variation since your traits will be different from your parents. So it can be a good thing. You may get something unique, and it's actually a big proponent to why evolution happens because it adds some variance into the population that can then be better than or worse than what's currently there. It can hopefully be passed on if it's good, and hopefully it dies off if it's bad. So, very key. So, that is protein synthesis. If you have any questions, make sure to ask. Bye.